All right, so this is our I Wonder project, me and Aiden's.
So today we're going to talk about bioinformatics and how we're going to use this throughout our project. So it actually allows researchers to look through sequence databases to find gene variations that could possibly lead to diseases or bacteria or pathogens. Uh, so far it is really easy to access to, especially now in our generation because time is accelerating.
Yeah, so in our kind of project, we considered two different questions. And so one of them was, how does real-time quantitative polymerase chain reaction, or qPCR, overall help messenger RNA and DNA sequencing throughout the collection of data? And now knowing about like the different types of PCR, which we did lots of research on, um, do we think that qPCR would be a good independent variable in our experiment? And so we're going to kind of talk about our experiment and then touch on our answer to that question that we got from our evidence. And then the other one was, how can we use DNA sequencing during this time of new social distancing and stuff because of the COVID-19 virus to help students and researchers still be able to learn and study DNA sequencing and the art of it. And because we're in such remote and at-home environments, can mail in DNA sampling and then gene databases that, just like Aiden said, we have more access to in the modern world, can it become more common? And why? Really, why is what we kind of looked at. Why should it be common among scientists and students? So for our experiment, we more did like an investigation and kind of made this skit where we took three made-up patients, James, Jay, and Delilah, who are college students, and then they live in dorms, and they were all showing different symptoms. And we gave them backstories, and they all had some sort of connection to each other. And they were admitted into the college infirmary, and we kind of looked into using DNA sequencing and gene databases, how we could find out what virus that they had, and then look at how tracing back to kind of how it could be transmitted between them.
So our first patient, his name is James, and he just recently had been admitted to the infirmary, and he told us that he was really fatigued and he had a massive headache, and he said that he had symptoms of such as like a cold, and he wanted to be home for the holidays, but he doesn't want to give these symptoms to his girlfriend Elena. He thinks that if he got just over-the-counter medications, it could be taken care of before the symptoms actually get worse. And James is Delilah's lab partner for an AP biology class. They have no known history prior to being assigned for each other for partners for a lab project. And they spend about 4 hours together 2 times a week, and they become close friends. Even though that eating food is not permitted during experiments, they share food and drinks during breaks and in between experiments. We also have our patient Jay, who comes to the infirmary and shows signs of a severe headache and a sore throat. He was out with friends and at a rave and believes that this soreness is due to him sort of screaming and shouting enthusiastically. However, he does request that he receives medicine for his headache. He has been awake every day this past week studying for his AP literature exam, but he does make some time to do some dancing, and he also starts getting involved with some dancing groups on campus, and he's taking a break in the waiting room. He spots James, who is also his neighbor in his dorm.
And then our third patient is Delilah. So Delilah was also admitted to the infirmary a while ago. She submitted blood, urine, and lymph fluid samples for testing and was later discharged. She felt awful. She was complaining that her neck hurt, and she just thought that it was because she was sleeping in the wrong position. She was planning to leave for a cheerleading competition 2 days from now, and she hopes that whatever illness she has will not prevent her from participating. So she goes back to the infirmary only to receive the results from the test that she took when she was admitted last time. So they were collected in the first visit, the blood urine test, and kind of the biology department in the college was working with also the molecular biology department to identify what pathogens were inside of her samples to see what was the cause of her symptoms. So they had to look at her DNA sequences. And the lab isolated some primers, which is, we'll talk about the process of actually DNA sequencing. And then they attached it to the genes in bacteria and viruses and amplified that sequence of DNA. And then they sent out Delilah samples, and then they compared the two. And so we're going to talk about how we will analyze Delilah's sequence and how we do that kind of at a remote at-home location. So this is Delilah's sample at the bottom you could find with thymine as red, adenine as green, cytosine as blue, and then guanine as black. And so we use this sample and we put it into a database, which we're going to switch over to and kind of explain the processes of going through it. And when we made the sequence, we used a random generator to make her sequence. But if we were looking at kind of the real world and not doing this in such a quick time, there's actually companies out there who take DNA samples that were mailed in from a remote location, wherever you want to be, and then you mail them into these companies, and then these companies actually sequence them for you. And so a name of one is the Sequencing Center or GeneScript, and they take your mailed in samples, they sequence it for you, and then they give you back information on what your base pairs basically. And so we then can take those base pairs, put them into the databases that are available to us, which we talked about. It's a new world with all of this technology. And so we have a lot of access to kind of the entire human gene database. Relating the slide back to our first question where we talk about quantitative polymerase chain reactions and how could it be used as an independent in our experiment, the answer to that would be yes, it could be used as an independent. It's not something that we would be able to actually perform since we're doing this remotely and not in the lab, and we don't have access to that equipment. But if this were to be done with mail-in sample, to mail into those companies that I just referenced beforehand, where you can mail in a DNA sample, if we had mailed in Delilah's sample, those scientists would have used the qPCR or another form of PCR, but qPCR is commonly used among scientists in genes, in DNA sequencing. They would have then used this reaction, and it would be the independent variable because Delilah's base pairs would be that dependent variable. It is what comes out of that reaction. And the reaction stays the same. It's still the process of the denaturization, the annealing process, and then the extension process. It's where it breaks apart that DNA strand. You bond a primer to it, like what we said, where we indicate the primers, and then it extends alongside one of those sides of that DNA, and it replicates it. And so that's kind of how the scientists get ample amounts of those DNA sequences so that they could find the base pairs. So yeah, it could totally be used as an independent in the experiment if we were doing this in the lab. And so we could take that and we could just look at it and compare it to sequences of pathogens and diseases, and that's what we did with Delilah. So now we're going to switch over to the actual site that we use to compare Delilah's sequence to virus samples.
So what we're going to do is we're actually going to go to this nucleotide blast section, and this is also the National Center for Biotechnology Information. So I actually have Delilah's gene sequence copied and pasted, so we're just going to copy and paste it into this accession numbers. And we're going to leave everything the same. The standard database is going to be the same. What we're going to do is we're going to do somewhat similar sequences, which is the blast end site, and then we're just going to hit blast because we're just inputting her DNA sequencing here, and just hit blast, and it's going to refresh for a couple seconds, and then she's going to explain what happens next.
So then after we click the blast button, then it's gonna give us a page that kind of shows the results of Delilah's sequence. So Delilah's sequence was 180 nucleotides long. So at the top of the screen, you could see the query length and it says 180. And then it displays, there's another tab called the graphic summary tab, and it displays sequences found in this database that match Delilah's sequence.
So this is the graphic summary tab. It shows the sequences found in the database that actually matches like her sequencing. Each colored bar represents a matching sequence in the database, and the red identifies like the stronger matches while the black identifies the weakest match. And for this DNA sequence that we have, there is actually no black. There aren't any other colors other than red. So we can show that this is like the highest alignment, the strongest match. And you can see on the top it shows the key level of alignment. Going back to the descriptions tab, you can see view the sequences that match the query sequence. It shows on the top the most common, and the bottom just has like the least. But on the top you can see it says Neisseria meningitis strain, and also there's also an accessions tab that shows the identical sequences that each scientist have. So if you click on that accessions tab and you click on one of those like the little links, you can see that it shows you have access to additional information such as like the full sequence of this gene and a description of the gene and like the names of the scientists that also have contributed to the sequence. And the e-value or accept value is like the number of blast hits you would expect to see by chance. The smaller the number, the more likely the match is accurate and not just by random. And we also have the ident, which is the short for max identity value. It describes the percentage of each nucleotide from the sequence. In Delilah's sequence, 100% of the nucleotides in the sequence match the query length, so we can say for 100% certainty that actually Delilah does have Neisseria meningitis strain.
So basically, all in all, like what Aiden was saying, we can gather from this information that Delilah does in fact have Neisseria meningitis strain. And basically just some information on this kind of thing is that it's a bacterium that can cause meningitis, and then it can also cause other life-threatening diseases like meningococemia, which is a life-threatening sepsis. And so substances can harm the body's potential to respond to an infection. And so these can be spread through close personal contact, living together, like how they're all living in college dorms, coughing, sneezing, kissing, and then even sharing through sources of fresh water. And so we remember, if we look back to the kind of descriptions of these patients and how they all knew each other, James and Jay, they lived close by each other, and Delilah and James were lab partners, and they often ate near each other. And so, if it'll let me go back to our slide, we kind of just ended up putting our questions at the end of our slide presentation because we kind of wanted to kind of go back to them and explain why our investigation was kind of used to fuel our kind of conclusion to both of our questions. And so in the question that references DNA sequencing and how in this time of separation, like clearly because we are doing a group presentation over a Zoom call, we can still learn and study about DNA sequencing, and we can still do this. And this is shown because like we were just taking a gene sample and we were able to put it through these gene databases, and the one that we used, it showed us the similarity between this sample and samples and sequences of pathogens and viruses, and we were able to come to a conclusion that Delilah had that meningitis strain. And so it was really interesting and cool to actually perform that and be able to answer that question and say, yes, like it is possible. And it should be more common because it was quick, it was easy. And sending it in to those companies that are already made to do that gene sequencing for you, it's really fascinating.
Yeah, and for the qPCR, it's a basic technique that a lot of researchers and scientists use for bioinformatics, and something as simple and easy as this. It's really used like everywhere in the science field, and you need several instruments and kits. But in this sense, with remote learning, we can only do so much. So using this site, I think it's very interesting that students and teachers can both use this, and it introduces new technology to students, and it is actually a lot easier because it measures the amount of PCR in the enzymes, and it actually helps a lot because it actually copies the DNA and ensures us that we are actually getting the right information that we do have.
And so, really, the reason why we kind of asked that question is, I mean, our questions changed a lot throughout our investigation. I mean, I think we changed them, each of them, like twice. And we kind of, at first, we were looking at just how DNA sequencing just kind of works as a whole. And then we were like looking more into it. And then we kind of like looked at all the different types of all the different polymerase chain reactions. And we found qPCR. And it was just really interesting to really see how it's one of probably the most benefiting and the most easiest types of reactions to use when gene sequencing, DNA sequencing. And so we kind of asked that question because we wanted to have a pretty solid background on how that process really works. And even though our second question talks about how we do it remotely and talks about how it can be done not in a lab, we still wanted to understand what actually happens in that lab and the science behind DNA sequencing. So yeah, I think that is kind of that's our project.